Here we go. Asexual reproduction. When you put the letter A in front of a word, it means not. So this is not sexual reproduction where you have two parents. In fact, the definition, it's reproduction involving only one parent. And what that means is the offspring are identical to each other and the parent genetically. They are identical to each other and the parent. We have a name for an offspring that is identical to the original. Begins with letter C. It's used in sci-fi quite a bit. Clone. I thought cloning was a myth. Nope. Cloning is not a myth. It happens in nature quite often. For us to do it artificially. So, what are some advantages of only needing one parent to reproduce? Well, it's really fast. For humans, to make a new human takes 9 months. For elephants, to make a new elephant, I think the gestation period is around 2.5 years. Most of the time for asexual reproduction, doesn't take that long. It's quick. Not only that, you can produce a lot of young. It's very, very possible to have dozens of offspring at once, hundreds of offspring at once, yea, verily, even millions of offspring at once. And no need to find a mate. Okay. If you watch nature shows especially, you'll see, wow, a lot of animals' lifetime, heck, a lot of humans' lifetime spent trying to find that special someone. Well, here you don't need to do that. That saves energy. There are some disadvantages. Well, the first disadvantage is there's a lack of variation. That's a V in the front. There's a lack of variation in the offspring. Because the offspring are all identical, what that means is they are all susceptible to the same disease or the same poor conditions, etc. Not a problem if that disease isn't there, Nick. But if a disease comes along, that can be an issue. That can be an issue. Offspring may be the same, may all be in the same location. Typically, species that reproduce asexually don't get around much, which means you might end up with hundreds or thousands or millions of offspring all in the same location. Well, that means that you're competing for the same food in the same space. Just to give you an idea, though, of the numbers that we're talking about, let's look at a bacteria, say. A bacteria can reproduce asexually. So, or a cell. How fast can cells reproduce? Let's have this cell double every day. So day 1, we're going to start with 1 cell. How many on day 2? 2. Those each split. How many on day 3? 4. Those each split. How many now? 8. After 8 comes 16. This is called a binary geometric series. After 16 is 32. We'll see how far we can go without our calculators. After 32 comes then 256. Then 512. Then 1024, then 2048, then 4192, then, give you a hint, 8,000. 192 times 2, that's going to be the same as 200 times 2 minus 16. 400 minus 16. 384? Is that right? Double check. Yeah. Heck. Times 2. 16,768. What's a quicker way to do this instead of going times 2, times 2, times 2? Ready? What day are we? Oh, we're off by 1 day. I think I want 2 to the 14th. Oh, because on day 1 we had 1 cell already. So if I go like this, 2 to the 14th, that gives me how many there are on the 15th day. I use my exponent button. You guys learned exponents in math?
Yeah.
Okay. So let's skip some. How many on day 20? Day 20 is going to end up being 2 to the 19th. 500,000. 524288. How about day 25? Day 25 is going to be 2 to the 24th. 16 3 7s and a 216. 16 3 7s and a 216. 16,777,216. Point being, you know what, Ibrahim? Asexual reproduction can grow pretty fast. You want me to do day 30, don't you? Okay, it's going to be 2 to the power of 29. After 30 days, we'll have 536 870 912. 536 870 912. 536 billion 870 million 912 cells. And that's once per day. Some cells reproduce faster than that. So a mass of cells becomes visible to the human eye at approximately 1 millimeter in width. You guys can see down to about 1 millimeter fairly easily. And it turns out that's around 250,000 cells. When would this mass of cells become available? I think right around day 19, because there's 500,000, so you'd have half as many the day before. Little nerd trivia. So, what is asexual reproduction? One parent, genetically identical clones. Those advantages that I gave you, that makes for a good test question, multiple choice or written. Those disadvantages that I gave you, that also makes for a good test question, multiple choice or written. That's a little hint. That's my way of saying I like this question, like this question, like this question. You're going to see it in the future somewhere. We want to talk about 5, count them 5, types of asexual reproduction. Okay? 5 types. The first is what we call binary, let's even type this in, Mr. Duke. Fission, Fission. Type number 2, budding. I thought budding is something you're not supposed to do in line, Mr. Duick. No budding. No, this is actually a science term. Fragmentation. Spores. And the last one is vegetative reproduction. Let me zoom in a little bit so you can see these a little easier. Tensei, is that big enough for you to read? Yeah, because you're kind of in my back corner. Maddie, is it big enough for you to read, okay? Okay. These are the 5 main types. Each is slightly different. Now, the nice thing is, based on the English meaning of the name Ibrahim, we can kind of figure out how each of these works. So it's not like they gave them Latin names like mitosis and cytokinesis, where I better have to memorize them. Anybody know what the word binary means? What does the word binary mean? What does the word bicycle conjure up in your mind? What? 2. Okay, binary means 2. Fission means break apart. You know what binary fission is? Split in 2. That's what your cells do. But also amoeba and paramecium. Budding, I'll bet you they grow buds like plant buds, sort of. Fragmentation, they break apart, they fragment. Yeah. Spores, I'm hoping you know the word spores. You've learned some science. Spores is one. And then vegetative reproduction. Reproduction means reproduction. Vegetables, we'll talk about how that works. So at least Mason, they didn't pick stupid Latin names. I got to get, I've gone on my rant about how in physics we know how to name stuff. Here they didn't screw up. First type, binary fission. Single-celled bacteria or amoebas use this method. And amoebas are those one-celled organisms that you see in pond water and things like that. They are complete life forms. So in an amoeba, the parent cell replicates its DNA and then divides in 2. In fact, it's just like cell division in humans, but this time, for an amoeba, it's the entire organism, because that's all it is. That's the whole life form.
So you mean like there's interphase?
Yeah, all that stuff. Okay. Bacteria also reproduce this way. Again, this is why bacteria reproduce so quickly. Bacteria, it turns out, only have 1 circular chromosome. So they don't go through mitosis. It's a different procedure. There isn't prophase, metaphase, anaphase, and telophase. But I'm telling you, at the end of it, Brian, the DNA is duplicated, the cell divides. It's just done differently. Think I have a picture? Yeah. Let me zoom in a little bit. So bacteria have 1 long single chromosome. Here's a bacteria. They're very simple organisms. The DNA is duplicated and it splits. It just doesn't do it by lining them up in the middle. There's no away apart anaphase or anything like that. Okay. What are some characteristics of binary fission? Well, for starters, it's very fast. You can feel it in your body when you're getting sick. You can almost feel that sickness, those bacteria progressing their way through your body. All of you at least once have kind of experienced that. You start out with a bit of a tickle in your throat, and like 2 hours later, you're like, wow, that happened in a hurry. Okay. Another characteristic, they can mutate rapidly. Remember, mutate is when those base pairs get switched, either through substitution, addition, or deletion. This is how bacteria develop antibiotic resistance. They mutate incredibly fast. In fact, right now, we're racing to keep ahead of bacteria that affect humans. We have these antibiotics, but almost any time we develop a new antibiotic, it's got about 3 or 4 years before enough of the world's bacteria have mutated that they're no longer responding to that antibiotic. We've got to try another one. This is why doctors, when I was growing up, doctors prescribed antibiotics all the time. Now they're very reluctant to do so, because every time you prescribe it, you're taking the risk that you're creating a mutating bacteria that will be immune. We don't want that. So most of the time now, we say, oh, your health isn't in danger. We're going to let you just get sick and get better naturally, please. There's no mixing of genes. So, oh, by the way, this is why you need a different flu shot every year. It's because the flu virus mutates so rapidly. There's no mixing of genes, so apart from mutations, offspring are all genetically identical. Oh, I turned the page apparently. Binary fission. How can I remember what that is? Fission, a fissure is a crack, a split. Binary, 2, split in 2. Next one. Budding. This is for fairly simple multicellular organisms, in particular 2 main types. We call them hydras, Hydras, and sponges. Did I give you guys this picture in your notes? I think I did. Okay, so a bud forms on the side. Eventually, it grows into an entire organism. It breaks off and forms a new individual. This is not binary fission because it's not one cell splitting into 2 cells. This is one organism forming a bud and then a completely separate but genetically identical organism. Budding. Oh, characteristics. I forgot to include that. First of all, it can establish colonies in one place. But because especially sponges and hydras typically are underwater, and water moves and there's currents, it turns out buds can also, come on, Mr. Dude, let's just handwrite. Move away to start new colonies. There's very little to no genetic variation, so all of the individuals will be affected by the same problems. For example, with the climate change and the fact that oceans are warming up, a lot of these species are in trouble because all of them are going to be affected by that. So, binary fission, budding, next on the list, fragmentation. These are simple organisms. They are multicellular. Simple organisms. Matt, you with me? Yeah. You're not looking at your phone or anything, are you? No? Okay. Can you put your backpack on the table, off the table on the ground? Thank you so much. I appreciate that. Sea stars, which is the proper name for what we all call what? Starfish. The reason technically you can't call them starfish is they don't belong to the fish family. But feel free to call them that. I'll know what you mean. But sea stars, some plants. So if an individual is broken into pieces, some or even all of the pieces will regrow into an entire individual. You can break them up, and some of the pieces will grow into a whole new individual. I don't think I include. Did I include pictures for this one for you guys? Okay. So sea stars were eating too many shellfish in an area, so fishermen, not knowing any better, caught the sea stars, cut them up, and threw them back into the water to let them die, thinking the fish would eat them. What happened? What do you think happened?
They regrew.
Each of those fragmented pieces became an entirely new sea star. Yes, some of the small pieces got eaten by fish. Most of them did not. And so they dramatically, these fishermen, increased the population of sea stars. Suddenly they made the problem worse. Plants can do this too. If you take a piece of a plant, put it in water to let new roots form. So Eurasian Milfoil is a problem in the Okanagan because of this. It gets stuck to boat propellers from lakes in the US. People bring that same boat up to Canada. The piece of the foil, a piece of the Eurasian Milfoil flakes off into the water, takes root, and spreads. That's why if you ever go through the border, you'll see signs saying, boaters, clean off your propellers, clean off your boats, or watch for Eurasian Milfoil. It's an invasive species. Some pictures. Some types of worms, you can cut them up and you'll get 4 new worms. Okay? A sea star, if you break up, there's a good chance that this leg that broke up will grow itself into an entirely new sea star as well. And some plants, take a piece of a plant, put it in some water, it'll start sending out roots quite often, you'll get a whole new plant. Fragmentation. Some characteristics. Again, can spread very quickly, and it's very, very difficult to actually kill the species, because most of the time when you kill the species, you might think, oh, I'll just chop it up. But even if an animal comes along and eats that species, the animal might not have eaten every little last bit, and there might be enough of a bit left over to start growing again. Okay. Vegetative reproduction. Vegetables, strawberries, and other similar plants will send out, and we call them runners. Small, tiny plants that stay connected to the parent plant for nutrients, etc., until they get established, and then once they're established, the runners die off. So you have your main strawberry, there's the root in the ground, it's sending out runners, these will start to grow, these will start to put roots in the ground, and then eventually some of the runners will just vanish. Some characteristics. Well, first of all, the new plants are supported until they're established, until they grow up. A negative, they're very close to the parent plant, and so this can lead to competition for nutrients, water, etc. And if that happens, it can lead to the parent plant being less healthy because it doesn't have enough water and nutrients to maintain its growth, its health. Allowing cuttings to form roots can also fit into this category. So, give you an example, and it's too bad we're towards the end of the year, but this is something for you folks to remember next year at springtime in front of the gym. Say, what? I'll show you. So an example of vegetative reproduction, there's something called grafting, Grafting, grafting. Some plants, apple trees, for example, they can't grow roots from cuttings. So instead, what farmers do is they take a branch of the tree that they want and they graft it onto a plant that has a root system established. Let me make this picture a little bit bigger. Okay? So here's a plant that has a root system established. You cut off part of the plant with a root system. This upper part is from the plant that you want to grow. You match it in, you wrap it, you wax it, you seal it, and the nutrients from the bottom plant will start to grow, And this plant here, which could be an entirely different type of apple, could be a very different species, or species is the wrong word, could be a different flavor type of apple, it'll grow. It'll grow. So grafting is a type of vegetative reproduction. Characteristics. The added branch, we have a fancy name for it. We call it a scion. S-C-I-O-N. Has the benefit of a strong, well-established root system. Another advantage, grafted trees produce fruit within a few years. Growing from a seed takes much, much longer. Grafting can also control the size of a tree. So, for example, is of a dwarf variety, and you graft a scion from a larger variety, it turns out that root will keep the overall tree smaller, even if the added branch is from a large tree. Springtime, something to look for. If you notice the cherry trees, are they cherry trees? The blossoms in front of the gym in the little parking lot, those trees there, if you look close, when they all blossom, one of the trees has one branch that's a totally different color of blossoms. Somebody has grafted one branch of something else onto there. Something to keep your eye out for. Kind of cool. How? By the way, I don't know whether it was done deliberately and artificially. If somebody did that, grafting can sometimes also occur in nature by a weird fluke of circumstances. But this is also then how we get all these different types of apples. You graft different apples to different apples, and you do that for successive generations, and you can tweak the flavor, you can tweak the color, you can tweak the tartness. Apples. And other fruits as well. You okay? You good? Good. Spores. Spores. So a spore is a single cell that is able to develop into a whole new organism. It's very light. It's very light, so it can travel very long distances. Somebody shove that underneath there? Oh, give me a test. Okay. Why would being light help you travel long distances? How do you think spores travel? Yeah, wind. Okay. Some of them are quite impressive. They can survive very harsh conditions. In fact, some of them can survive, I kid you not, hundreds of years without starting to develop if the conditions aren't good. So, for example, if there is a drought, or if there is extreme temperature, it'll just sit there. But when the conditions are right, it'll often still be able to develop. Based on the picture, can you tell me at least one type of organism that uses spores? Okay, mushrooms and fungi. Yeah. Ferns do as well. Okay. Spores. I have a little summary chart here. Did I give you this chart in your notes? Okay. It's table 5.1. It's from your textbook. Advantages and disadvantages. Oh, I think we did this actually early on. I gave you advantages and disadvantages. So advantages, large numbers of offspring are reproduced very quickly from only one parent when conditions are favorable. You can get a population explosion. If you're a life form, that's a good thing. Large colonies can form that can outcompete other organisms for nutrients and water. They can overwhelm an area and choke out other life forms. If you're the dominant life form, that's a good thing. Large number of organisms means that species may survive when conditions or the number of predators change. They're really tough to entirely wipe out the species. Almost always a few of the population survives, and then they can reproduce again. And energy is not required to find a mate. Matt, there are also some disadvantages. The offspring are all genetic clones, so a negative mutation that occurs that can make organisms susceptible to disease or destroy large numbers of offspring. Some methods of asexual reproduction produce offspring that are close together and they'll be competing for the same food in the same space. And unfavorable conditions such as extreme temperatures can wipe out entire colonies. So, pros and cons. Okay, change gears a little bit. Cloning. These 5 methods of asexual reproduction are all examples of cloning, but they only work for fairly simple plants and animals. Complex plants and animals can only be cloned artificially in a laboratory, and even that we're not very good at. We can sort of do it. So we're going to talk about cloning. Some of the myths and some of the reality. Okay. We have this human-assisted cloning. Why? Well, to help save genetic information from an endangered species. Jurassic Park being the creepy horror movie example of that. Oh, let's bring back the dinosaurs. Or to mass produce an organism with a desired characteristic. Seedless watermelons. It wasn't from nature, we made those. One that we're working on now, we're trying to clone skin cells. We would like to be able to, if we can, make new skin for burn victims. A couple that we're working on that I hope do come true in the future. Well, oh, hang on, let's go like this. We're hoping to be able to grow new organs. We're getting close. Right now, we use transplants. If you have a kidney issue, we need to transplant someone else's kidney. If you have a heart problem, we have to wait, sadly, for someone else to die but have their heart survive the accident. We transplant the heart. We would like to be able to actually grow these, especially if we can grow it from your own cells. Then you remove the risk of your body rejecting the transplant. Your body would recognize it automatically. Treating Parkinson's disease. What's Parkinson's disease? Does anybody know what Parkinson's disease, guys? You guys know what Parkinson's disease is? Michael J. Fox had Parkinson's disease. Generally, Parkinson's disease strikes older people. Michael J. Fox got it much younger. I think he got it when he was 34, 35. Usually hits people in their 60s. Your body starts to shake. You can't control your muscles. Your body trembles involuntarily. Well, it might be possible to clone the nerve cells and take care of that, or to clone the brain cells and take care of that. Maybe we could grow a new spinal cord for someone who's paralyzed and transplant it. Or maybe we could cause their spinal cord to grow and fill in the gaps after the injury. We don't know. I hope so. Then there's another type of cloning called reproductive cloning. And the most famous one is Dolly the Sheep. Dolly the Sheep was the first large-ish animal that was cloned, that was made in a lab by combining two different types of DNA from a mother sheep and a father sheep, but not having it grow in a sheep's womb. Sorry, not having the reproduction occur naturally. We combined the DNA in a lab. Then we implanted it into a sheep's womb. So it says this: all cells have all the information needed for the entire organism, but they have forgotten how to use most of it. Your skin cell has within it the information required to make a brain cell. Your brain cell has within it, in its DNA code, the information required to make an eyelash cell. But all of these are switched off. So scientists took the DNA from an adult cell and they put it into an egg cell that had the nucleus removed. This egg was then implanted into a host mother, and Dolly was allowed to grow. Now, if you read this, what's missing is no DNA came from the father. We took the DNA from one cell, from one organism. We removed the nucleus, put that same DNA into the nucleus, which means the sheep that grew only had one parent. That means it's genetically identical. It's a clone. Put your pencils down. Look up. So, scientists remove, let's make this a bit bigger so you can see the picture. I think I took this from your textbook, but that's okay. Scientists remove the nucleus of an egg cell from a female sheep. Very tough to do because the nucleus is not very big. A mammary gland cell is removed from an adult female sheep. This cell and the egg cell are placed next to each other in a bath of chemicals. A jolt of electricity, yeah, right out of Frankenstein, causes the two cells to fuse. Oh, where's my step 4, Mr. Duick? There it is. The fused cell begins dividing to form an embryo, and then this embryo is inserted into the uterus of a surrogate mother to complete its development. The resulting lamb is a clone, genetically identical, of the sheep that donated the mammary gland cell. It's a clone. So, how did Dolly do? Born July 5th, 1996, but when I was reading this, Josh, this was huge news all over the world. This was the first time we'd actually cloned an animal. Cells is one thing. This was a big animal. Died young, but non-cloned sheep often die young too. There was a virus on the farm she lived on and several sheep died. At the time, there was no evidence that she was less healthy. However, lots of cloned animals are unhealthy or the embryos die very, very early. It's been tried many times. It doesn't seem to work very often. And we think animals that are cloned, they may have weaker immune systems, maybe? Is that why they die off? So we did a sheep. Then along came Snappy. 2005, about 10 years later. Snuppy, not Snappy. Snuppy, a dog. Put your pencils down, look up. So here was the process that was used to make the first cloned dog. They made 1,095 eggs from donor mothers and the DNA from ear cells of a 3-year-old Afghan. These were implanted into 123 surrogate mothers. 3 of the pregnancies took. 1 had a miscarriage. 2 went to term. The other clone died when only 22 years old, but Snuppy made it into adulthood. Why would we be interested in cloning dogs? Why would we be interested in cloning dogs? Well, for starters, wealthy people who want their pet back. Their pet dies, they want the same pet back. Clone it. That's one. Other reasons? Well, think about it. Pick your pencils. Nearly done. I know it's been a lot of me talking this time. You good? You okay? You sure? Yeah? Okay. How much more am I covering, Mr. Dewick? Let's go like this here. Let's pause for a sec. So we'll pause here.